Welcome to episode 1 of module 8, Non-Infectious Diseases and Disorders, looking at the first inquiry question: how is an organism's internal environment maintained in response to a changing external environment? Our syllabus reference for this video will construct and interpret negative feedback loops that show homeostasis, including temperature and glucose. Our learning intentions for this video will define homeostasis, outline the stimulus response model, distinguish between positive and negative feedback loops, and interpret negative feedback loops for glucose and temperature. Homeostasis. Homeostasis is defined as the process by which organisms maintain a relatively stable internal environment despite changes in the external environment. What does this mean exactly? We say an organism is in homeostasis when there is a balance of gas concentration, this is oxygen and carbon dioxide, nutrients, intake of carbohydrates, proteins, lipids, as well as vitamins and minerals. Ions, this can be sodium, potassium, magnesium, and chloride ions that are essential electrolytes for electrical neurality in cells, generating action potentials in the nerves and muscles. Water balance, as well as maintaining optimal temperature at 37 degrees Celsius and maintaining appropriate pH depending on the location in the body. When any of these become too high or low, chemical reactions within cells cannot occur efficiently. Think back to Module 1, enzymes. These chemical reactions are called cell metabolism. When this happens, it may lead to diseases and even problems in the human body. For example, diabetes is a condition where the body cannot maintain glucose levels. There are two stages of homeostasis. The first is detecting changes from the stable state, and the second, counteracting these changes. This is where the nervous system has an important role to play in the coordination of all other systems that work to maintain homeostasis. The stimulus response model. The stimulus response model is a diagram that shows the interactions between the stimulus, receptor, the central nervous system or endocrine, the effector, and the response. Let's look at this in more detail. Stimulus, a change in the environment, either internal or external, that is detected by a receptor. Receptors are groups of cells in the body that detect the changes in an environment, called stimuli. Two examples are thermoreceptors that detect changes in temperature and chemoreceptors that detect changes in the concentration of chemicals. The central nervous system is made up of the brain and spinal cord. Its job is to receive the transmitted information, interpret it, and send signals to act upon this information. Effectors are parts of the body that work to counteract the change. They include muscles and glands secreting chemicals. The response is the organism's reaction to the stimulus. The response is the organism's reaction to the stimulus. The central nervous system transmits the response to the effectors, which counteract the change. Feedback loops. Homeostasis is maintained in the body through positive and negative feedback loops. A negative feedback loop simply reverses the change. For example, if your body temperature is too high, a negative feedback loop will act to bring it back down towards the set point of 37 degrees Celsius. A negative feedback loop can be illustrated as a chart or an annotated sequence of diagrams. For a complete picture, homeostasis will usually involve the interaction of two or more negative feedback loops. The first loop is stimulated when a parameter like body temperature is above the set point and the loop intends to bring it back down. The second loop is stimulated when the parameter is below the set point and the loop intends to bring it back up. Positive feedback involves a response that reinforces the change detected. It functions to amplify the change. A change is detected by a receptor and an effector is activated to induce the same effect. This promotes further change. Positive feedback loops will continue to amplify the initial change until the stimulus is removed. Examples include childbirth. The stretching of the uterine walls causes contractions that further stretch the walls. This continues until birthing occurs. Lactation. The child feeding stimulates milk production, which causes further feeding. This will continue until baby stops feeding. And ovulation. The dominant follicle releases estrogen, which stimulates the LH and FSH to release and promote further follicular growth. Glucose and temperature negative feedback loops. On a really hot summer day, our body temperature is going to increase past its set point. This increase is detected by thermoreceptors on the surface of our skin. These thermoreceptors will transmit the signal to the brain, specifically the hypothalamus, where it will send a message to the effectors. For example, the hypothalamus may send a message to our sweat glands, signaling them to increase sweat production. Our blood vessels will often dilate and direct higher blood flow toward the skin in an effort to dissipate any excess heat. As these effectors are taking place, our body temperature begins to decrease back to homeostatic point. On a cold winter's day, our body temperature may decrease past its set point, especially if we aren't wearing enough warm clothes. This decrease in temperature is again detected by thermoreceptors on the surface of our skin. These thermoreceptors will transmit the signal to the hypothalamus, where it will send a message to the effectors. Our blood vessels will constrict and move the blood flow away from the surface of the skin to the organs in an effort to keep our bodies warm. Often, when vasoconstriction is occurring, there's usually a change in the colour on the surface of your skin. Shivering will assist in generating heat in the body, thereby increasing blood flow and warmth in the body. As these effectors are taking place, our body temperature begins to increase back to homeostatic point. Normal blood sugar levels in a person who is not diabetic ranges from 3.9 millimoles per litre to 5.6 millimoles per litre. When blood sugar levels increase, the insulin secreting cells in the pancreas detect the change. The pancreas produces insulin. Insulin triggers liver, muscle and fat cells to absorb glucose where it was stored. This will then bring the blood sugar concentration down and back to homeostatic point. When blood sugar levels decrease, the glycogen secreting cells in the liver detect the change. The pancreas instructs the liver to break down glycogen and release glucose. The response is blood sugar levels increasing back to homeostasis. And that is the end of episode 1. Thank you for watching.